So unlike prokaryotic cells, eukaryotic cells are compartmentalized. What this means is that structures in the eukaryotic cells allow for specialization. So we have organelles such as the mitochondria, such as the nucleus, which you can see outlined down here in this cell where the DNA is stored. We have mitochondria, some mitochondria down here at the bottom left, all the way along the bottom here. In plant cells, we obviously then have chloroplasts, and there are numerous other organelles, which are compartments found in the eukaryotic cell. And this allows for specialization. That means that specific biochemical reactions can occur in particular parts of the cell, but not in others. So, thinking about specialization of cells, on the left here we have some neurons, and you can see they have a cell body and then long dendrites and axons. And then on the right we've got epithelial cells, which are flattened cells, the purple structures with a little tiny nuclei here. Now if we took these neurons on the left from your brain and these epithelials on the right from the inside of your cheek, they would have the same genome, and that's because all of these cells were derived from the fertilized egg. On the other hand, they've got very different proteomes, and those proteomes give the cells different structures and therefore different functions, and that's come up in the past, so we won't dwell on that right now. So the bottom line here is we've got the same genome in these cells, the same individual, but different proteomes, and it's the proteome which is responsible for the differences in structure and in function. So what we're going to do now very quickly is we're just going to run through the major structures that you find in eukaryotic cells. So we'll talk a little bit about the cytosol, and then the cytoskeleton, the flagella and the cilia, the endomembrane system, the mitochondria, and lastly chloroplasts. So this is a quick overview of cell structure and function. So cytosol is obviously found in all cells. If you're a cell, you've got to have cytosol. Also known as cytoplasm, and for us the two can be used interchangeably. The cytosol is a major site of the breakdown of molecules in the cell, so-called catabolism, and so many reactions of cellular respiration occur here, but also there are many reactions which build molecules up, and so those are called anabolic reactions. And so this is the major site of catabolism and anabolism. That's not to say that these processes don't occur elsewhere in the cell, it's just a defining feature of the cytosol. And the cytosol also coordinates a lot of activities in the cell, so when information is received by the cell, that information is processed by molecules within the cytosol, and a lot of cellular responses are elicited from within the cytosol. And so the cytosol is the main site of catabolism, anabolism, coordination. And there are numerous other functions which are too detailed and too specific for us to get into. Also in the cytosol, you find translation, that is the synthesis of proteins using the information in messenger RNA molecules, and so that happens in the cytosol. The cytosol is also the home to the cytoskeleton. We'll talk about that in a little bit more detail in a few minutes. So the cytoskeleton is the home is made of three structures: microtubules, the intermediate filaments, and the actin filaments. And in your textbook there's a table which compares and contrasts these three structures, and so it's probably a good idea to be able to compare and contrast these yourself. So we'll look at each of these in turn. We'll start off with the microtubules. Microtubules are a very nice example of quaternary structure. That means that they are made from the assembly of smaller individual proteins, and the two proteins that are used to assemble microtubules are this protein called alpha-tubulin and this protein here called beta-tubulin. And so alpha-tubulin associates with beta-tubulin, so you get a dimer, that's one alpha-tubulin and one beta-tubulin, and then those dimers assemble into this long filament that you see here. And if you were to look down from the top or from the bottom, you would see inside of the microtubule is a space called the lumen. So this is a long cylinder, and the microtubules have a plus end, and it's on the plus end that the alpha-beta tubulin dimers add to the microtubule to make it grow, and so these grow and shrink, and so they grow at the plus end and they shrink at the plus end, and then the minus end that's usually anchored, and in animal cells that is anchored at a structure called the centrosome, and the centrosome is made of a pair of centrioles, and so that's where all of the microtubules originate from in animal cells. And in plant cells there are no centrosomes. There is what is called the microtubule organizing center, and so that's where all the microtubules originate from in plant cells. Intermediate filaments are so called because they're intermediate in diameter when you compare them with microtubules and microfilaments, and so they're kind of in between, about 10 nanometers in diameter, as you can see here. And these are like twisted rope-like fibrous structure, and they have this staggered alignment, and so you have an individual filament protein here, and that gets wound around another filament, and then these form tetramers and higher order structures, and then they get wrapped up to make rope-like structures. So I'm not getting into a lot of the functions here. I'm going to leave you to take a look at the functions of these structures in your textbook. And lastly, in the cytoskeleton, we have actin filaments, also known as microfilaments, so you may hear them referred to as microfilaments. These again have a plus end, and they have a minus end, and like microtubules, they grow at the plus end. Actin filaments, as you'd expect, are made from a single protein called actin, and the actin polymerizes into long chains, and then those chains wrap around one another to form a 7 nanometer diameter fiber. So that's the microtubules, the intermediate filaments, and the actin filaments. And if you look at these in cells, you can see different distributions. And so as what you're looking at here are falsely colored electron micrographs of some cells. So on the left, we're looking at the microtubules in green, and the DNA is in blue here. And you can see that all these microtubules originate from the centrosomes, which are right here. So even the ones which are down here, what looks like to be in the bottom of the cell, are originating from this point here by the nucleus, and they obviously go around and back and behind or in front of the nucleus. And so they're distributed in this fiber-like network throughout the cell, and that's related to their function. The intermediate filaments in the cell, you can see they're not as organized as the microtubules, and they're shown in green here, and the nucleus would be right around here. So this is one type of intermediate filament we're seeing. There are many, many, many types of intermediate filaments. And then the actin filaments you can see here in orange and red, and you can see these make very rope-like structures. And again, they kind of look like the microtubules, but they're more rope-like and fibrous in structure. So that's the distribution of these elements of the cytoskeleton in the cells. Now, the cytoskeleton doesn't really do too much. At least some parts of the cytoskeleton don't do too much without the help of a group of proteins called the motor proteins. And these help move things around in the cell. So we're looking at here a protein called myosin. And so myosin is a protein, it's a motor protein, and it's got 3 main components. It's got the tail over here on the left, it's got the hinge, which is kind of here in the middle, and it's got the head. And so myosin has this tail domain, and that will bind to other structures in the cell. It could be a part of the cytoskeleton, it could be some vesicle carrying neurotransmitters and a neuron, that kind of thing. The hinge is exactly what it says. It's a flexible region that can bend. And then there's the head down here, and that will bind to one of the parts of the cytoskeleton, maybe a microfilament or a microtubule. It also binds ATP and hydrolyzes the ATP. And as what you'll see in a moment is that when that happens, you can get the hinge region here to flex, and that can create movement. So what we've got here is we've got a myosin protein right here that is attached at its head end to a actin filament. We can see these 2 strands of actin molecules. Another nice example of quaternary structure in proteins. And so the head binds to the actin filament. It releases, and the head changes, or the hinge cocks. And then when we get rebinding in this 3rd step to the actin, we get ATP hydrolysis, and we get the bending of the hinge. And so what you have is this head that is basically binding, cocking, releasing, rebinding, cocking, and as it does that, it can walk along the actin filament here. And so anything that's attached to the tail up here would be getting dragged along this actin filament by the motor protein. So here's another example of a motor protein. This is kinesin, and what we have here is we have some cargo. This could be like I said, a vesicle, a neurotransmitter-filled vesicle. And here, kinesin is this double-headed motor protein. So here's the head. There's another one here. It's on the other side, the microtubule, and we've got these two kind of tails which are twisted around one another, and they bind the cargo. And so as the heads cock and release and bind, and cock and release and bind, the motor protein walks along the microtubule. And there are some really nice animations of this behavior in Canvas, so I'd really encourage you to watch those animations. So that's what kinesin does. So kinesin works with microtubules, and myosin works with the actin filaments. So let's go back to our myosin example. One of the things that myosin does is it works in muscle contraction. So we have an actin filament again here, and here's a whole bundle of motor proteins. And what we can do is we can tether these tails to another actin filament. So there would be another actin filament along the top, bound to the tails, and then the heads are bound to this actin filament here. And so when we hydrolyze ATP and we cock the hinge, this region here, we can cause the two actin filaments to slide over one another, sorry, and that's what happens when you get muscle contraction. You can also use these motor proteins to cause cytoskeletal structures to flex. So here we've got some bundles of microtubules. So there's two microtubules here, and this is the positive end, two over here. And if we anchor the microtubules at the negative ends, the minus ends down here at the bottom, now they can't slide like actin and myosin, but what can happen is when the motor proteins on the left here, these heads, when they try and walk along these microtubules over here on the left, and when they're attached to these microtubules on the right, because the microtubules are anchored at the bottom, the motor proteins can't walk, and what happens is we get bending or flexing of the microtubules. So rather than sliding of the microtubules past one another, we get bending and flexing, and that can be really important in generating movement in things like flagella and cilia. So, like I said, there's plenty of movies in the Motor Proteins Canvas page, so check those out. Plenty of cool stuff to see there, give you a better idea of how those motor proteins work. They're very dynamic, so very difficult to talk about without seeing some animations. Okay, now let's move on to kind of more middle-ish of the cell, I guess. And I'll talk about the nucleus and then the endomembrane system. So the plasma membrane is on the outside of the cell, and the endomembrane system is on the inside of the cell. So endo means inside. So this is a membrane system that you find inside cells, and it functions with, but is independent of, the plasma membrane. So the endomembrane system is composed of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the plasma membrane, and then a variety of different types of vacuole and structures which are derived from the endoplasmic reticulum or the Golgi. So that's what we'll look at next. Okay, so let's just start this off by looking at the structure of the nucleus very quickly. So we're looking at the nucleus here on the top left, and if we blow that up, there are some important structures to look at. We've got a nuclear envelope, which is surrounding the nuclear material. So bear in mind, this is like a sphere. So this is 3 dimensions, and we've cut kind of like a quarter of that nucleus away, and we're seeing inside the red stuff is the DNA associated with proteins, which is collectively called chromatin. And then we've got the nuclear envelope around the outside, and embedded within the nuclear envelope are these pores. These are structures which allow materials to move in and out of the nucleus, and so you can see this is blown up down here. So this piece here is the nuclear envelope, and there's another piece of nuclear envelope down here, and this structure here is the nuclear pore, and this complicated structure, which you don't need to worry about too much, basically regulates the movement of material into and out of the nucleus. So that's the basic structure of the of the nucleus. Let's now start thinking about the endomembrane system. So the endomembrane system really starts with the nuclear envelope, and then moving away from the nuclear envelope we have endoplasmic reticulum. And there are two types of endoplasmic reticulum. There is the so-called rough endoplasmic reticulum, and the rough endoplasmic reticulum is rough because it has these ribosomes dotted all over its surface. So that's the hallmark of the rough ER, and it generally is closer to the nuclear envelope than is the smooth ER. And so the smooth ER is biochemically very different and functionally different than the rough ER. It lacks ribosomes and does some different functions. So I'm going to leave it to you to read in your textbook the functions of the rough ER and the functions of the smooth ER. So I'll leave that to you. So we've got rough ER and smooth ER. These membranes kind of form like plate-like structures called cisternae. And so the cisternae are these flattened kind of tubules. So this is a cisternae here. It's like a flattened tube. So if you took a tube, a cardboard tube or a sheet of paper, you rolled up into a tube and then you squished it flat, that would be a cisternae. And so the ER, the endoplasmic reticulum, is made up of these flattened stacks. Inside of the ER lumen, so all the stuff that you would see in here, that space there is called the ER lumen. We'll come across a lot of lumens. We've already seen one. The microtubule has this space inside called the lumen. And that's where a lot of the biochemistry and the physiological reactions of adding sugars and storing calcium and detoxifying toxic stuff, a lot of that stuff happens in the ER lumen. And if you're curious about what this looks like through an electron microscope, here's an electron micrograph of some rough ER, and there's a little bit of smooth ER in here as well. A little bit of smooth ER, a little bit there, a little bit here, and a little bit more down around here. And so that's characterized by no ribosomes. All these red dots are ribosomes, and so this is an electron micrograph, and it's been pseudo-colored, it's been falsely colored. The nucleus is up at the top here, and then this structure down here in the bottom right is a mitochondrion, and we'll talk about that in a little bit. And so that's what the ER would look like if you could see it down in an electron micrograph. So as we move away from the nucleus, we have a structure called the Golgi apparatus, named after an Italian scientist. And so one of the functions of the endoplasmic reticulum, shown over here on the left, just as rough ER, is to send proteins that get made on the ribosomes in the rough ER to the Golgi. And in the Golgi, there is a lot of biochemical modification of those proteins. So we get the addition of sugars and that kind of thing. And so those proteins move from the endoplasmic reticulum in these little vesicles. So a vesicle is just a sac of membrane, and so this is just a little sac of membrane which is pinched off from the endoplasmic reticulum, and it's going to come over here and dock. So it's going to fuse with the Golgi, and then another vesicle will leave up here and end up fusing down in the next stack of the Golgi, also called a cisternae, and another vesicle will leave and come into the next stack in the Golgi, and another one will leave. And so as these vesicles move through the Golgi, more and more proteins get modified by the addition of sugars in a process called glycosylation, and many of the proteins which are released from the cell are very highly glycosylated. And so ultimately here we've got a little vesicle which has left the trans face of the Golgi, and that contains a number of molecules and proteins which are going to be released outside of the cell. And so when we look at the structure of the Golgi, the side of the Golgi, this side here of the Golgi, which is closest to the endoplasmic reticulum, is called the cis face. Then there's a middle section called the medial section, you can see that down here, and then the side which is closest to the cell membrane is called the trans face. So you can remember which face is closest to the ER. That's the cis face. That's where material arrives from the ER, and then the material moves through the Golgi via these vesicles, getting progressively more and more modified until eventually material leaves from the transpace of the Golgi. So this is a secretory pathway. Now, not everything that enters the Golgi enters and leaves destined to be secreted like we're seeing here. Some materials will be packaged into vesicles and actually kept inside the cell. Other vesicles will actually leave the endoplasmic reticulum and never go to the Golgi. They may become something like a peroxisome, for example. So some of the vesicles which leave the Golgi, which don't actually exit the cell, end up being structures called lysosomes. And lysosomes are for degrading parts of the cell which are no longer functional. So proteins that have been broken down or that need to be broken down because they've stopped functioning, they can be taken into the lysosome and broken down by a group of enzymes called the acid hydrolases. And these enzymes only work in the low pH of the lysosome. So remember, if it's at a low pH, there's a high proton concentration. And so what these enzymes do is they will take a molecule. It will take a molecule, and that's just being shown here as a group R1, which is just some piece of a molecule attached to another piece of the molecule R2. And in the presence of water, they will break the bond, and that's a hydrolysis reaction. Hence, these things are called hydrolases. And so when we use water to break this bond here that I've indicated, we end up with one piece of the molecule here, R1, and then another piece of the molecule here, R2. And so you can probably guess that if you wanted to link these two pieces back together again, you could combine that hydroxyl on the left and that hydrogen. And if you combine that, then you would get a water molecule back. And so that's perfectly reversible. It's not going to be reversed in the lysosome. The point of the lysosome is to break these things down. Just a quick recap. Some of you have seemed to miss the idea that if we skip a quiz because of a holiday, the material from the previous week just gets added to the week in which we have a holiday. So that means that your next quiz is covering 2 weeks, and those will be the learning outcomes from weeks 3 and 4. So that's important that you keep on top of that kind of thing. So the last thing that we should talk about are the chloroplast and the mitochondria. And I'm not going to share any slides on this because I think that the chloroplast and the mitochondria get beaten to death in high schools and in classes in community colleges. So there's some very simple reading in your textbook you can do. I will just summarize very quickly the structures and functions of the mitochondria and the chloroplast. Let's start with the mitochondria. The mitochondria is a membrane-bound organelle. It's got an outer membrane and an inner membrane. And the inner membrane contains a substance called the matrix. And between the two membranes is a compartment called the intermembrane space. And we'll talk about these structures in much, much more detail when we talk about cellular respiration. All you need to know about the mitochondria right now is that its function is to synthesize ATP, and it's also the location of the Krebs cycle, which we'll talk about when we talk about cellular respiration. So right now that is good enough. Just supplement that with some additional reading from your notes, from your book, as needs be from the learning outcomes. The chloroplast, a little bit more complicated in structure. There are 3 sets of membranes: an outer membrane, an inner membrane, and then a stack of membranes called the thylakoids. And so inside the inner membrane is a substance called the stroma, and then inside the thylakoid is the thylakoid lumen, and we'll talk about that when we talk about the reactions of photosynthesis. Now, the only thing you have to really know about the function of the chloroplast right now is that it's to build sugars. And a lot of people seem to think that the function of the chloroplast is to make ATP. No, the chloroplast does make ATP. That is, that's required for the chloroplast to make sugars. So the goal of the chloroplast is to make sugars, and the goal of the mitochondria is to synthesize ATP. And in doing so, it's actually breaking sugars down. So that is really a very, very quick tour of the cell. A lot of this is very basic stuff, so don't need to spend too much time on it. use the learning outcomes. They will direct you to what you should pay attention to in your readings. And the last thing I'll say is make sure you pay attention to the verb in the learning outcome. If it asks you to explain something, that is very different than describing it. The learning outcomes text on your screen is grey and orange, or maybe black and red, depending on the way you have your monitor's colour set up and how you're watching this. So that's a description of the colours. If you have to explain the colours, that's very different. That's all to do with reflection of certain wavelengths, absorption of other wavelengths, and how light is perceived by the different types of cells in your retina. So make sure you discriminate between define, describe, explain, list, calculate, evaluate, compare and contrast. So make sure you pay attention to those verbs in the learning outcome, and that will set you up for much greater success on your quizzes. That's it, folks.